Good day, Matrix, and welcome to today's lesson. I'm Renee Bishop. I'm a subject advisor for Life Sciences in BCM, and today I'll be presenting paper 1 to you. Please make sure you have a pen and paper on hand to take notes, and don't forget to write your questions in the comments section, and we'll get back to you as soon as possible. So let's get on with today's lesson on paper 1. Another question that is very commonly asked in our exam paper is about a control or a baseline. Now, a control and a baseline are two different things. So let's clear up what each one means. A control is a group in an experiment that does not receive the treatment of the independent variable. In other words, it does not have the independent variable that we are being tested. In our previous example of the rats that received microplastics in their water, the control group would be the group that does not receive the microplastic, because microplastic bioaccumulation was the independent variable. Now, why do we have a control? A control is there to compare with the other experiments or groups to show that it is the independent variable, right, that is causing the effect and not any other factor. Okay? So, and control is important to show that our results are correct and the results are due to the independent variable. And there's no other factors in our experiment that are affecting these results. So remember to learn your definition for a control. It is the group in the experiment that does not receive the treatment of the independent variable. And then also remember to learn why a control is needed. Okay? Because it is to compare and show that it is the independent variable that causes the effect and not any other factor. But now, when we have a specific example in our question, for instance, our example of the rats whose male fertility we are testing, we need to answer this question specifically. So, in the case of the rats in your trial paper, the reason we had a control group was to show that it is the microplastics. Right? We say that it is the microplastic bioaccumulation okay, that caused the effect. The effect in this case was the decrease in fertility there we go, and not any other variable or any other factor in this case, and not the water that was given with the microplastics. Okay, so we answer the question specifically according to our example given in our question paper. Right, so remember to replace the highlighted words, right, with the correct example in your question paper. Now, how is a baseline different to a control? A control group is a completely separate group of subjects or samples that do not have the independent variable, whereas a baseline, all right, is the same group, but the baseline was measured before the treatment was given, okay, and so the recording is without the treatment of the independent variable. An example of this would be when we are giving somebody, for instance, a food diet, and we want to see how glucose level is affected by eating food. Before the person has food, we would take their glucose level. This would be the baseline measurement. And then we would take their glucose levels after eating food. The reason we take that baseline measurement before they eat the food is so that we can compare and show that it is the food that is affecting the glucose levels, all right, and not other factors that might affect that person. Okay, so a baseline is the same person or sample that we are using, okay, before the treatment, whereas a control is a completely separate group to those that we are doing the experiment on. Right, and then let's just quickly wrap up, okay, our scientific investigation, right, and we're going to look at graphs and the common mistakes that we make in graphs. Firstly, be very careful to read what graph is being asked for and draw the graph that is asked for in this investigation. Many times we see learners drawing a pie chart or a line graph when they're asked to draw a bar graph. The caption of your graph should have both the independent and dependent variable and how it is being measured. Your caption should be a complete explanation of what is being shown in the graph. Only draw what is being asked for. Sometimes you are only asked to draw certain bars and not all the data that was given in the table. You are going to lose marks if you draw all the data when only specific data was asked for. The bars of a bar graph and a histogram should be the same size. Please make sure you have a ruler in the exam and that you use this ruler when drawing your graphs. Many times we see learners that are drawing the graph, the bars skew, and they're losing the marks because the bars are not the same size. Also ensure that you draw the scale correctly on the y-axis. Many learners lose marks for incorrect scale. And then there are a number of calculations that you need to learn for your exam. Okay, so firstly you need to calculate the difference. Okay, difference means you are going to subtract. You need to calculate percentages, right? Whenever you are calculating percentage or percentage increase, ensure that you write times 100. In most question papers, you get 1 mark for just saying times 100, even if the value before is incorrect. So it's worth just writing times 100. Don't leave that question out of your paper. Right. Then you are also needing to learn how many times greater, okay, and also to calculate the average as well as calculate ratios. Make sure that you go over these calculations and practice them from past papers to make sure that you can do them. And that brings us to the end of our session. Okay, I hope that you've gained some knowledge from the session and that scientific investigations will no longer be as scary to you. Looking forward to seeing your results at the end of the year. Good luck with the exams and study hard. Thank you.